Okay, so this chapter is on lipid and amino acid metabolism. Last chapter was on carbohydrate metabolism. So as we go through, we first need to know what blood lipids are. And blood lipids actually are basically phospholipids that have turned so that their hydrophobic end is inside and their hydrophilic end is on the outside. We've got triglycerides in the center, cholesterol, esters, and we've got these alloproteins that sort of go through the layers and help us out here. So I'm going to give you a minute to write that down.
Whoa, we're starting the test already?
Yep. You have until Friday to get the other tests done. But we just don't have a lot of time to play with. So we do a little bit of lecture, then we'll go back to lab. Sorry, Gio. We'll put this up. Hopefully it will be up. He's at AP Psych test. And we hope we didn't ruin his test. It should be in the... It's the yeast lab. Let me know when you've got all that filled out. Got two more still writing fervently. One more. Okay, Amber, I'm going to have you catch up with somebody after if you can. Okay. All right, so we're going to listen to this little quickly thing. Okay, so how do we metabolize fat? So have you ever looked at chicken and you've taken it off and looked at the fat layer? It's sort of fatty. That's your adipose tissue. And that's this muscle, this part right here, this yellowish part is your adipose. And that's what I was talking about. You needed to go to the adipose or the skeletal or the heart is where we'd find it. And this sort of gives you an idea of what kind of things could be used for. Okay. So this is, if you ever watch the doctors, they also show you when they open up people, it is yellowish fat that's inside. And a lot of people keep that adipose tissue around their waist. That's why as they get fatter, that's where it's stored. Okay, so this sort of shows you, what I was just telling you. I forget the order that I have these in. So fat mobilization, the hydrolysis of stored triglycerides followed by the entry of fatty acids and glycerol into the bloodstream. So we've got the liver, the kidney, and the gallbladder, the colon, and the small intestines that help us to metabolize things. And as we go through our cycle with our liver and our heart and our brain and our muscles and what they need, adipose tissue can help us get back glycerol, and fatty acids can be turned into ketones and triglycerols. Now we have to be careful. If we make too much ketones, we end up with ketoacidosis, which I think we talked about last time in last chapter, or it's in this chapter at the beginning. Can't remember. Very low density LDL. A lot of people talk about their HDL and their LDL, but we're adding in there also very low density lipids, and we're also adding intermediate. So those are a couple things that we are adding. We okay to move forward? Okay, so this is looking at your liver. And if you have a fatty liver, that adipose tissue is actually attaching to the liver. So that is a bad thing because your liver's not working as well as it should. And we've got several things that are going on. Amino acids are to build muscles. We've got glucose that we need. We've got many different cycles going on within the liver. And so if the liver is damaged in any way, it makes it really hard to live because this system is dependent on others. Remember that they said they are water hating, so their hydrophobic end goes inside. That's why it makes a perfect circle. Oops. So when we get to glycerol metabolism, we take glycerol through ATP and the enzyme glycerol kinase. These names are just horrible. We're ending up getting SN-glycerol 3-phosphate. Then through NAD+, we can, and the glycerol phosphate dehydrogenase, we can get dihydrogen acetone phosphate. And that is the process of glycolysis. We get our phosphate again, our PI, moving through. Okay. And this becomes a very important pathway for us. Okay to move forward? All right, so beta oxidation process. This is also one that you need to make sure that you understand and know how to do. This is a pathway in which the fatty acids are broken down into molecules of acetyl-CoA. This page in your book, page 782, is going to be very, very important for you. So I did not put it in your notes, but it's something that you need to go back over. Okay, so 782 in your book. And this one basically, every time that it goes through this cycle, it loses 2 carbons. So if I have a chain that's 18 carbons long, it has to go through this cycle 9 times to break down. Okay? And every time you get to the end here, it'd just be 2 less on the chain. And this is one of the problems that you're going to need to be able to do. So just know when I go through a beta oxidation process, whatever the length of the fatty acids is on top, by the time I get through 1 of that, it's going to be 2 shorter. Okay, and it goes down till you just have 2. How many times you have to go through it. So I'm just going to let this run real quickly. Pop this back on. So most of the time you are starting with a fatty acid, so you have to activate it to start with. So you have to give up those 2 ATPs to start with. Okay. I'm glad that he did do that cut down because you actually do have to only go through that cycle 8 times because the last time you go through, you split the 4 into 2 2s. So just remember, you can divide it by 2, then take away 1. But that does give you the number of acyl-CoAs. So I hope that makes sense. I liked how he did that, so that's why I put that one up. Okay, so then we got ketone bodies. So on ketone bodies, we need to know these 3 ketone bodies. You need to know them very, very well. Okay? So I'm going to turn the light back on so you can see what you're writing. You go just a bit, about 5 more minutes of lecture, and then we'll go to lab. So with these ketone bodies, you need to know where they're coming from too. So I don't know if I have that on this slide or not. So ketone bodies can be exported to the as energy sources for the heart, muscle, kidney, and brain. And they're specific to it. I must have it on the next slide. Let's see what we got on the next slide. Oh, okay, I might have it later on. We'll see. So ketonemia is an elevated level of ketones in the body, in the blood. Ketonuria is the presence of ketone in the urine. And acetone breath is a condition in which acetone can be detected in the breath. So oftentimes it smells fruity. although some people say it smells like nail polish remover, and that's what acetone is. So, but I've heard it been described as fruity too. So if you look at the name of it, ketonemia, elevated in the blood, ketones, ketouria, that's in your urine, and acetone breath, of course, is when acetone is detected in your breath. Okay? All right, so ketosis versus ketoacidosis. So we need to know the difference between the two. And I found this chart and I really liked it because they're not the same. So ketosis is a condition in which ketonemia, ketouria, and acetone breath exist together. Okay? So you have a low level of ketones in the blood, you have normal process in the body, and you have safe function of low carb ketogenic diet. So this is a lot of people that went on the Atkins diet ended up with this because all they were eating was protein. They didn't have any carbs and they didn't have any lipids. They had some lipids, but they were basically eating meat. Ketoacidosis, key here is acid. So this is a low blood pH due to elevated levels of ketone bodies. Extremely high levels of ketone in the blood. So difference, ketosis, low, ketone high, ketoacidosis. Can turn the blood into acid, and it's deadly if treated. You think of your blood running around in your blood vessels all of a sudden turning to acid, what's going to happen? It's going to eat those away. This occurs in diabetics when they don't take enough insulin or aren't well. People are starving or alcoholics is what the bottom part is. So this happens when people aren't well, very well, or they have been fasting too long or starving or are alcoholics. Okay, does that help you make the difference? Everybody okay with the difference? And the thing here is just remember acid. This is all acidy, and this is osis, meaning low. You got normal processes in the body, safe function of a low carb diet, but you got to be careful. All right, going forward. So what is ketoacidosis? Early symptoms, it's just diabetic stuff. So frequent urination, thirst, very dry mouth, high blood glucose, high levels of ketones in the urine. So oftentimes when you go to the doctor, the first thing they test is they take a urine sample and they want to see what's going on. If you have high levels there, then they go to the blood glucose and go from there. Then if you get farther along and they don't catch it early, some other symptoms are they're always tired, nausea and vomiting, hard time breathing, fruit odor on your breath, dry skin, really dry skin, and itches like crazy, confusion, they're not sure what did you just say? They just can't figure out what order is going on. So, acidosis is a low blood pH. Some symptoms there. We've got an optimal ketone zone that we want to keep going in. We've got what happens with starvation, ketosis, and if it goes too far, it goes to ketone acidosis. We've got a nutritional ketosis that starts over here. So we can be at either side for ketosis, but ketoacidosis is when we get really bad. All right? So fatty acid synthesis. So we start with amino acids. We put the glycos in. We do pyruvate. And from pyruvate, we go down into the PDH, the acetyl-CoA, the citric acid cycle, and this is happening in the mitochondria. In the cytosol, all these other things are happening. And we end up with oxidation, desaturation, and we're working with NADPH again. And all these names should be starting to come familiar if they're sort of all going mishing together. Okay, I'm going to do one more slide, I think. Okay, this one is fairly simple. Amino acid metabolism, I didn't say that right. Metabolism, thank you. I'm not saying that right. Okay, so we have an amino acid pool. An amino acid pool is used to synthesize, to get energy production, to synthesize glucose or fatty acids, and to synthesize non-protein molecules that contain nitrogen. And we have what's called protein turnover. So either we are taking this pool and we're making body proteins, or we're breaking that body proteins up so we can get energy. And our body tells us what we need at what time. So it'll decide, yeah, you need some more body proteins because you're building muscle, and we need more collagen, we need more actin, we need more things that are holding you together, or this set is getting old, we need to redo it. Okay, I'm going to stop us right there because I'm looking at the clock, and I want to make sure we have enough time back in lab.